action. So CASP has been kind of a testing ground for future Nobel laureates, right? So we had Michael Levitt as an assessor some time ago, and in the morning we heard from John Jumper, and now we have David Baker. So he got his recognition for protein design, but obviously he started as a protein prediction guy. And David will correct me, but I believe it was CASP-3 when he came to CASP for the first time. And since then, David's name was always on the top of all the lists of winners in CASP. And believe it or not, it's my second time pleasure to introduce David. So first time we had this milestone CASP experiment, which was CASP-10, and we invited people who are, names in the field to give talks. And then I remember it was Gaeta, and I was sitting before introducing David somewhere in the hall, and there were two students behind me, and one student asked the other, okay, so that's the slot where David Baker should talk, and then David comes to the podium, and the other student says, oh, he sent his graduate student to talk. David looks so young as he is looking now. So David, so without any further introduction, so the stage is yours, please go ahead.
All right. Well, thank you very much, Andrei, for the introduction, and yeah, thank you, and John Christoph for inviting me. We were just discussing comparing the weather here and there, and I also feel kind of bad. I'm probably keeping you from the beach, but yeah, I mean, I think long before we started working on protein design, we were thinking about protein folding and protein structure prediction, and I'm just incredibly indebted to John and the whole CASP team just for the CASP experiment, which was really really key for me getting started on my independent career here at the University of Washington, because at the time, as everyone knows, at the time that John started CASP, there were papers coming out saying that the protein structure prediction problem had been solved, and it was really hard for new people to be able to evaluate what they were doing, and it's hard for people just to get started and show they had some ideas that were worth pursuing. And I think CASP really made that possible, and so I probably wouldn't have gotten to design if it hadn't been for CASP. So in this, I dug up this image from a long time ago on the left, on an old school Rosetta simulation, folding simulation. This is sort of what we talked about, and this was the big thing in CASP-3 and CASP-4, and it's now so outdated that it's been sitting in some computer file for the last many years. But anyway, so I'm not talking about that today. Today I'm talking about de novo protein design, and so we've been developing. So we developed Rosetta first for structure prediction, and then we realized we could go backwards to protein design. Brian Coleman made top 7, and then we've really been focused on designing proteins with new functions ever since. And then with John Jumper's demonstration of the power of deep learning for structure prediction, with AlphaFold, we figured we had to learn how to do some deep learning stuff. So developed some deep learning methods for protein design, RFdiffusion for the generating backbones, protein MPNN for assigning sequences to backbones. And then, of course, we can use AlphaFold or RosettaFold, and now 5 other methods for predicting the structures of the design proteins and see if they match the intended design model. And so the way that RFdiffusion works, just very briefly, it's inspired by methods for image generation, diffusion methods. And so to train these sort of methods, you take all the images off the internet, noise them to different extents, and then train a network to remove the noise. And then once you've done that, you can start with a completely random image, random collection of pixels, and progressively denoise it to get something that looks like an image from the internet, a member of the training set, but not. And in the same way, we took the PDB and noised every structure at different extents, trained a network to remove the noise, and Once we had done that, we could start with completely random distributions of residues, progressively denoise to generate a structure that looks like it could be from the PDB, but it wasn't. So just in the same way that you can condition image generation on a text prompt like a cat sitting on a table, you can condition the protein diffusion process. And so, for example, you can condition it by saying, by providing a target, in this case, the insulin receptor. And in that case, the diffusion process, it senses the receptor as it's building up, and it's been trained where some part of the protein of the system is kept fixed, so it knows about interfaces. And so we get structures out which are very shape complementary. And so basically, what I'm going to show you for the rest of my talk is basically applications of this kind of design, mostly diffusion, a little bit of old school Rosetta stuff. And I'm just going to show you examples from different areas. And in each case, after we have the design on the computer, we make a synthetic gene, or generally we're testing quite a few designs. So we make synthetic genes. We express them in bacteria usually. We purify the protein out, and then we measure to see whether it does what it does. And then what I'm going to be telling you about are just different applications of protein design. And in every case, I'm going to be showing you sort of a design model and then experimental characterization of the design protein. So I'll first talk about design of binding to protein targets. So I showed you that example with the insulin receptor. Here is the TNF receptor that's involved in inflammation. Probably a number of you have taken drugs which disrupt this pathway, antibodies. So we were interested in what would happen if we tried to diffuse a protein against the TNF receptor. You can see we get these really shape complementary proteins out that kind of hug the surface. We can optimize these designs by a little bit of partial diffusions. That means adding noise and removing the noise. It's kind of like a general refinement approach. And we get down to very, very high affinity. These designs can be targeted. We can target different members of the family in this way, which is interesting. Many of them are therapeutic targets. And I should say that these proteins also turn out to be quite effective at blocking inflammation in animals. So there's a lot of interest in the pharma industry in designing antibody-like structures. And so here we just basically condition the design process on giving it not only the target, in this case, the influenza hemagglutinin, but also we specify that it should be an antibody, or in this case, a VHH nanobody-like fold. And what comes out are these is the cryo-EM structure comparing the design model to the actual structure. And you can see, like, we're not specifying, how the VHH should come in or what the conformation of the loops are. And you can see they're coming out really quite accurately. We can also make binders to disordered proteins and peptides. So this shows binders to the A-beta peptide and the a peptide from tau. And you can see what happens here is that they're sandwiched in between in the middle of this extended beta sheet. And if you our collaborators in Thomas Knowles' lab in Cambridge basically carry out aggregation assays where the peptides added to solution, it aggregates. That's what's being measured on this axis. And then if you titrate in the design protein, it basically shuts down the aggregation. And if you add enough stoichiometric amounts, you completely shut it down. We can also make binders to just kind of arbitrary protein peptide sequence. And so this is work by Keija Wu, where she basically took many different peptide sequences or parts of proteins that were disordered proteins. And what she's doing here is building these proteins with these extended grooves where the peptide fits in, and then kind of the amino acid side chains can get read out. And these turn out to be really selective. And so this is the design binder against the design peptide targets. And you can see that they're, for the most part, very selective for their targets. And this is kind of neat because it gives a way of targeting intrinsically disordered proteins, which has traditionally been a hard problem because they don't have a defined structure. And here, there's actually a crystal structure comparing the design model to the crystal structure of the design model. And the nice thing is that we're kind of forcing the disordered core protein to induced fit bind to this in this or extended arrangement where you can read out all the amino acids. So we can also design membrane proteins. So this is a collaboration with Anastasia Vorobyeva's group in Belgium. And so we can, so there's a lot of interest, obviously, in beta barrel nanopores for sequencing and other things, but that's all been using naturally occurring nanopores. And here you can see these are, we can design beta barrel nanopores of 10, 12, and 14 strands. And as the barrels get bigger, the pores get bigger, as you can see here. And we can get these inserted into lipid bilayers, and so the conductances go up. So this is kind of a neat way we can now sort of fine-tune pores for different types of applications. One thing that we can do is Lena Anna has been working on methods for designing small molecule binding proteins, and this is showing this is a design binder to small molecule cholic acid. Again, the superposition of crystal structure and design model. So what she and several others in the group did was to fuse this binder at the top of one of these designed nanopores. And then it's really cool because you get a gated nanopore. When the small molecule is added, the nanopore conductance shuts down. And so it's a way of making sort of very selective sensors. And I'm kind of excited about the general possibilities of making kind of molecular electronic nodes out of sort of designed gated channels like this. And Alexei and Jinwei, working in collaboration with Jens Gundlach's lab, have been making completely sort of pores that are kind of foreign to biology. Again, they're being made with this diffusion process, and they're being designed to fit into pores drilled into silicon wafers, silicon nitride. And the idea is that then with silicon, you can get a much higher density than with lipid bilayers, and you can also have more direct coupling to electronics. So for sensing applications, for sequencing applications, there's some really exciting advantages. And this is, of course, something that biology doesn't do, which is, use pores that insert into silicon nitride, for example. We can use the same diffusion approaches to generate DNA binding proteins. And this is, and we've been using sort of this type of diffusion approach as well as traditional Rosetta methods. This work is led by Cameron Glasscock, who's just starting his own job at Rice University. And this shows what kind of comes out. So you have the DNA duplex, and then you have your specific binding protein, design binding protein up here. It makes very specific side chain base interactions. And if we measure the activity, the binding activity, we take basically the DNA duplex with the sequence, and it binds the target. And then these red boxes show what happens if you mutate away the residues, the bases that are interacting with the DNA binding protein, and the redder it is, the more activity is lost. So these are really quite specific for their intended targets. So we've been also, for a number of years now, designing proteins that self-assemble into different types of three-dimensional architectures. In this case, this one was is a icosahedral structure, and my colleague Neil King during the pandemic put the spike protein or the receptor binding domain of COVID-19 on it, and this worked really well. It produced a really strong immune response, and this has now been approved for use in a number of countries. Now, we've been wanting to go larger.
David, can you hear me?
Yeah.
We
Yes.
for whatever reason it is.
Oh. Can you hear me now?
Went out. Stepan, projector went out. Okay. We are restarting projector, sorry.
Okay, all right, just tell me when. And did you, should I just stay on this slide?
No. It was fast.
Okay, okay, all right, all right. So I was just describing the vaccine, and the everything we had designed up until recently was completely symmetric. So if you think about an icosahedron, you can sort of also view it as a dodecahedron, and every vertex on a dodecahedron is identical. But the problem is, if you want to make it bigger, like we've been thinking about delivery applications, you have to break symmetry because there's no other regular platonic polyhedron a regular polyhedron that larger than the icosahedron. And so the way we're familiar with that happening in nature or in just normal life is like on a soccer ball, you have your pentagons, but you intersperse them with hexagons. So hexagons will just tile a totally flat plane. And what you can do is insert as many hexagons as you like between these pentagonal vertices here and make much larger objects. But the problem here is you have to break symmetry because now now the different placements are not identical. And so like this one here has actually, let me see. So the well, you can see that at this point here, two subunits are on the edge of a pentagon. One subunit is on the end of a hexagon. So there are two different ways that we've been playing around with to solve this problem. One is to explicitly design proteins that have some symmetry. So if you look at this trimer here, this is a cryo-EM structure. You can see these two edges are on a pentagonal face. This edge is on a hexagonal interface. And this is pretty big. This is AAV, which is for scale, which is a a common delivery vehicle. And the other way you can do it is by putting different protein chains on the different surfaces. So here we have these blue and green ones, which are on the pentagonal surface, and then the orange ones, which are on the hexagonal one. And so both of these work. This is also a cryo-EM structure. So we can now sort of this field has advanced to the point now where we can go beyond the limitations of strict symmetry and make more complex objects. And so now the question is, how do we make these non-porous and get them to deliver contents? Everything I've shown you so far are proteins that have single low energy states, but we can also now design proteins with multiple low energy states. And so this builds on the work of Florian Pretorius and Phil Long, who designed systems like this, where we call them hinges for obvious reasons. They're in one state until an effector comes along, and then they go into a different state. And Arvind Pillai realized he could use this to make allosteric assemblies. So here we have a square-like structure that, and when you add the effector, what you predict happens is these bend, and now the overall geometry of the structure should change, but it's not happening through a direct, the effector isn't at the interface, it's far from the interface. So this is basically an allosteric effector that's working in sort of this rigid body allosteric way. And Arvind constructed a number of really neat materials. Here's one that goes from being this square-like tetramer, and then you add the effector, and it goes into a this pentameric 5-chain compound and or material. And here's one that goes from being a 3-chain trimer to a 4-chain tetramer. Arvind even sort of emulated what hemoglobin does. Here's a design which has 4 effector binding sites, and the oligomerization state doesn't change. Its shape clearly changes. And one of the things that was remarkable about hemoglobin when it was first discovered was how cooperative it was. And that's true for Arvind's design system as well. As you start adding in the effector, you only see unbound material and then fully occupied material. So if you look at sort of how much bound state there is as a function of the concentration of effector, it's very sharp, and it's just It's got a Hill coefficient of nearly 4, so it's as cooperative as hemoglobin for oxygen. And actually, I think, well, I'll just show you. This is we can also allosterically control kinetics. And so the basic idea is that if you have a bimolecular complex and you want to compete it with something else, you generally have to wait for the interaction to be broken. But what Adam Broerman figured out was ways of actually facilitating the off rate. So here the effector binds, you get this allosteric change, and that drives off the this bound partner. And indeed, this is experimental data. As he adds in this effector, you get greater and greater off rates. Now, where this sort of becomes interesting biologically is Adam took a protein that we had previously designed that binds the IL-2 receptor beta and gamma receptors, and this is a potent activator of signaling. And he built in the same kind of inducible steric switch if I go back and forth. So this is up, but now when a second protein is added here, it knocks off the second receptor subunit. This works really well. I'll just show you, this is the same kind of acceleration of dissociation. But we collaborated with Jacob Peiler's group in Germany. They do these really cool experiments where they have a cell labeled with the beta and gamma chain of the receptor in different colors. So you add this molecule, they come together, and then you add the effector, and you can watch them come apart. And in fact, they come apart very, very fast. And if you look at signaling, this is when you add the blue molecule, signaling goes up, and it would just keep going up unless you add the effector, and then you can shut it down. And this is interesting because there's been a lot of speculation that the thing that's important for cytokine signaling is how long the receptors stay dimeric. And so now this gives a way to actually test that directly. And just to show you some applications outside of biology, here is a designed protein made by Nate Ennist, which holds 2 chlorophyll molecules in exactly the arrangement that they are held during photosynthesis in the photosystem. And so now we can isolate sort of the key part of the reaction center in a soluble protein. And then we can start building it into these larger assemblies. So in this case here, these cubic structures, which have this special pair chlorophyll binding protein. Each one of these blue things is here. You can't see the chlorophylls, but they're there. And then they're held together in this larger cube. So we're very interested in building artificial light harvesting systems. And then finally, we've been using these methods to design enzymes. And here the idea is what you condition the network on is the structure of an active site you want. So you specify the active site residues and where you want the substrate to be, and then you build a protein around there. And so Anna Lelko and Sam Pellock applied this to the classic reaction of ester hydrolysis. And so what's neat is you can basically choose what you want the active site to be, and you can make it increasingly complex. So this is a very simple active site with a histine and a serine. This is the classic catalytic triad out of a biochemistry textbook with a single oxygen hole. And when Sam and Anna made these kind of designs, they got things that were activated nucleophiles where the serine could react with the probe, but nothing actually happened. And so then they added initial glutamate to back up the histine, and now they got enzymes which were single turnover enzymes. And what was happening was that the acyl group was piling up. That this reaction proceeds by an intermediate. The serine was carrying out the first step in the reaction, but then it was stuck, and water couldn't release the acyl group. And so then they found that if they added a second oxyanin hole stabilizing group, then they could get full turnover with quite respectable overall rate. And what's neat is, these are small, these designs are small proteins, so they're relatively easy to get crystal structures of. And so here you see the full catalytic triad here, and you see the serine, the double oxyanion whole, and so in traditional enzymology, you kind of solve the structure and then you zoom in at the active site and you sort of make up a story about how it works, but you can't really move around where the functional groups are. And whereas with this kind of build-up Novo design approach, you can really test hypotheses and explore what's key for catalysis. And here, this is now looking at metalloenzymes. So this is starting with a density functional theory based active site description, where you have histines that hold a zinc ion that then activates a water, which attacks the carbon, which then takes the place of the serine in the previous example. And here, this is work from Donghu and Seth. They were able to get in the first batch of designs they tested, they got this a design which really stood out. It turned out to have a kcat over km of 22,000 per mole per sec, which is starting to approach the activities of native hydrolases, which have evolved for millions, or in some cases billions of years. And so this is exciting. Now we're obviously trying to go for reactions where there aren't really good natural enzymes, for example, for plastic and other polymer degradation. And I should just say we've been developing ML-based methods for that that can sort of model ensembles, because one thing that an enzyme really needs to do is to bind its substrate in exactly the right conformation and place the catalytic side chains around it. So this is, and we use this post facto, applied it to our designs. It turned out the most active design, which we had actually labeled A1, we labeled that because it had a really pre-organized site, was much more, once we knew the activities, we found that the less active designs had much less pre-organization, whereas the active designs were quite pre-organized. And then a final example. So in nature, we have many examples of tooth and bone that where it's proteins interacting with inorganic ions to mediate mineralization. And so Amihai and Harley, whose names got dropped off here, unfortunately, set out to do this with an unnatural chemical, zinc oxide. And so they designed proteins which were sort of complementary to the zinc oxide lattice, and then incorporated them into these nanocages. And when they grew these nanocages in the presence of zinc oxide, they found that some subset of them had these sort of darker regions, which corresponding to zinc oxide mineralization. And then inspection of the structures. So in this design, the cage here has it's an octahedron. It has both threefold and fourfold axes. The zinc mineralization surface is along the threefold axis, and indeed, when the cryo-EM structure was determined, there was this extra density right here, corresponding to which we think it corresponds to the zinc oxide, whereas the fourfold axis was always empty. There was nothing on it. So the zinc oxide is getting deposited right adjacent to the designed interface. And so that's kind of a whirlwind tour through protein design now. I think it's really exciting because there are so many different applications. And again, just want to thank John and the whole CASP team for helping me get started and get into this area, which I think really required some ability with protein structure prediction just to be able to evaluate designs. Because once you've made designs on the computer, how do you test them? Well, the first way you can test them is by predicting the structure to see if the design sequence actually is predicted to fold to the design structure. And that was actually the biggest use we had for Rosetta in the early days, was sort of using it as a filter to evaluate our designs. All right, well, that's what I wanted to tell you about, and happy to take questions.
Thank you so much, David. Very exciting talk, and we have some time for questions. Good 10 minutes, so let us go. Who wants to ask first question? John.
Hi, David. John Walt.
Hey, John.
So it's really amazing how far this stuff has come, and it seems to be sort of exponentially growing. What do you see as the limitations? What are the remaining problems? What needs to be solved?
Yeah, well, I think it depends on the area. I think in medicine, which is obviously a big area, the problem is I think we're getting a little bit beyond the biochemical function. So I think design of binders is now approaching being a solved problem. But as you know, going from a binder to a drug, there's still a lot of steps. And a lot of those things are just as hard as they were before. So, as the problem is there, if we had a huge database of clinical trials, all the ones that have been carried out with failures and successes, then you could maybe train a model that could make things that be better past clinical trials, but those data sets don't really exist. So I think the biochemical function problem, I think, is becoming more and more solved is probably too strong a word, but we see how you can sort of see where it's going. I think the big questions are for medicines, the stuff that comes after the binder. For materials, what are, how do you get out there? I'll tell you what we're working on now, which I'm really excited about: molecular machines, which couple. I talked about binding and I talked about catalysis, but if you put together a catalysis, so you have a cycle going around with binding to different things as you go around the cycle, you basically get a machine like a Walker or a disaggregase. I'm really excited about this this ability to template inorganic mineralization. I said a little bit about. There's a lot of opportunities for enzymes, and think about enzymes that work on DNA for editing. So I would say that the biggest challenges are, okay, we've got these powerful tools. What do we apply them to? And then, how do we go for these higher order functionalities like combining binding with catalysis or with delivery? I guess another thing that we're doing a lot of work now on is conditional binding. So you want something that will only bind to a cell and, say, poke a hole in it if it's gotten certain markers on itself. So it's sort of combining these base functional modalities, I think, is sort of a current frontier.
Thank you.
Okay, please introduce yourself and ask question.
Hi, I'm Patrick Bryant. So you said binding is kind of a solved problem, but when I look at these methods, they use hollow structure as input, right? And in a lot of cases, you need to kind of specify the binding sites. So I wonder, how do you know that this is a solved problem for new targets where you don't have such information?
Oh, well, let's see. We've designed binders to many, many, many proteins where we're targeting sites where there's nothing known that binds. I mean, yeah, and I think it's now working. You can use predicted structures as well. So I mean, I think there's still challenges like really charged surfaces. It's just what you think. We can look at a surface and if it's got some hydrophobic residues, then that's easy. If it's all highly charged, it's much harder. So yeah, but these methods have been validated over and over again on sites. Like, for one of the things we've been doing is designing proteins for cellular trafficking, where you specifically want the binders to bind to the cell surface receptor in a place that doesn't overlap with the natural ligand, so it can just get taken up and direct lysosomal degradation or something.
Sure. But I just mean, I don't know if this is unpublished data but I haven't really seen a large set of new target interfaces that doesn't have binders been designed to, right?
Oh, yeah, we're actually just trying to write a review on sort of trying to collect. I mean, the problem is the binder stuff is all kind of spread around, but thank you for pointing that out. We'll definitely highlight where the interface is different from what's been from anything that's known. But there's definitely quite a few cases.
Thank you.
Hi, David. Great talk. Susan Sudakawa. You
Hi.
I had a question for you as you're writing your review. When you're doing these diffusion models and creating these binders, how much of the interface is to or using main chain?
Oh, well, I think it would vary. Like we just posted a preprint actually where and we were struggling with polar targets, and one of the things that we found is if for proteins like with IG folds that have beta sheets, if you target an edge beta strand, it's a really good way because you can use an edge beta strand in your design, and so then you satisfy all those polar groups in the backbone. So that's turned out to be a very effective way to make binders to relatively polar things, is to specifically target beta sheet, exposed beta sheet hydrogen bonding things. And then, of course, in Helix, you don't have much, but in loops, there's sometimes, yeah, we should look at that. But yeah, there is a class of binders where we're preferentially, I mean, you need to have interactions also that give you specificity, but yeah, targeting beta sheets is a good way to go.
No, I was just wondering for your diffusion models where you're not rationally designing it, does it pick up those strands?
Yeah, it will, so we can cheat. I mean, so we can either let it just go free, but sometimes what we do is we say, okay, target these residues along the beta strand. So so sometimes like, yeah, earlier today I was talking to someone in my group who's been testing out the latest diffusion models, and they have this predilection for making extended beta sheets. So so you can certainly get models that just unconditionally really prefer those sort of interactions.
Okay, thank you.
Hi, thank you for a really nice talk. It was amazing to see all the incredible advances with deep learning for the design problems that you've been working on. I was wondering what your thoughts are on if there are any problems that you don't think will be solvable using deep learning, and that we might need other strategies for, physics-based or other approaches.
Can you please?
Yeah, well, I think the general answer to that is for things where there aren't many examples in the protein structure data bank. I mean, all of the advances in protein structure prediction and in protein design really they're leveraging what's in the PDB. So, so for design with really exotic unnatural amino acids or using enzymes with unnatural cofactors, or, for that matter, in catalysis, we're starting with, say, a quantum chemistry model of the active site. So I think when you start going beyond the 20 amino acids and really, and particularly when you start including, types of interactions at the atomic scale, which aren't represented in the PDB, then I think that's where deep learning methods start running out, and physically based methods are really useful. So we still use the Rosetta-Sorval atom modeling for systems where there are unnatural amino acids and cofactors.
Thank you.
Chandra, you're asking a question, or okay, you're just moving. All right, so David, it looks like that's it with the questions. So again, thank you so much for finding time for us this evening. I believe it hectic for you these days, and congratulations from all of us, like from the whole field, for your great achievement, and take an extra day going to that Stockholm event, because Stockholm proved to be a difficult destination.
Well, enjoy the beach, and yeah, it's nice, and thank everyone there. Thanks particularly to John and the CASP team for all your work over the years, because it totally directly led to all of this. So
thank you so much for your nice words.
Yeah. All right, thank you.
All right, it was great talk, and now we are off to the next section, which will be a discussion on protein and QA. So we have organizers of the session. So are you going on stage, or you are staying here?